Hello, good morning. So, following the molybdenum, we just slowly moving towards the thing where you have the tungsten in the biology. So, today we will start little bit where we were last time, and slowly we will move to the coordination chemistry of tungsten in biology. So, what we have seen basically that how the molybdenum coordination environment is helping us to show some important catalytic activity. So, the coordination environment is very much important, and that environment showing us something which is very much related to the corresponding electron transfer behavior of the center, because all these cases where we see something for their catalytic activity which is very much dependent on the metal ion, which is bound to the enzyme pocket and its corresponding oxidation states, because these catalytic activities related to molybdenum is very much dependent on the electron transfer behavior. So, we just wanted to know what sort of thing is basically controlling the initially the thermodynamics of this electron transfer and afterwards the kinetics of the electron transfer. So, what we see last time that when we have the environment and when we have the selenocysteine system, that means the selenium atom is there, which is there for the Formate dehydrogenase family. So, if you have the Formate dehydrogenase family where you can have one selenium center and which is binding to or interacting to the molybdenum center, and this molybdenum center for the different catalytic activity is in +5 oxidation state. So, we have molybdenum in +5 and which is basically settling between two other oxidation states, which is the oxidized form, which is molybdenum 6 plus, and other is the reduced one, which is molybdenum 4 plus. So, what we find there that during this transformation for this electron transfer, so is a 2 electron transfer process when you go from molybdenum tetravalent state to molybdenum hexavalent state. It is not only giving something where the corresponding donor groups which is there available there can stabilize this center in +4 oxidation state, but also in the hexavalent state the other groups are interfering and which can stabilize the center in +6 oxidation state. So, for the controlling factor for all these things that you can have 2 E0 values for these two states, and these E0 values are basically can tell us like the very simple thing what we all know, that if we have some intermediate oxidation state like that of our, we all know that the cupra state has one oxidized state is the cupric state and another reduced state is the copper 0. So, it is well known to us from a very basic chemical knowledge that if you do not have some groups which are well suited, that means which are little bit soft, because the copper 1 center is comparatively soft compared to the copper 2 center which is hard. So, if soft groups are available which can stabilize the copper 1, then only we get this copper 1 center in the stabilized form. Otherwise, it can disproportionate between copper 0 and copper 2 plus, and here also like this you have two E0 values involving there for the process what we call it as a self reduction and self oxidation process. And this self oxidation and reduction process also tells us that whether we can have something, that means the corresponding disproportionation, and the reverse of that the conproportionation reaction involving three oxidation states, which one should be stabilized and which is there available. If something is also related, that means if the catalytically active form, if something is mediated for the reaction involving copper 1 and copper 2 and the copper 0, if some intermediate is forming as copper 1 plus. So, some physical methods should be available which can be utilized for the detection. So, the detection of this is very important. So, how we can detect the presence of copper 1 in the system? So, we can have some spectroscopic methods, we can have some other techniques. So, that depends on the corresponding properties. So, the physical properties of this particular oxidation state of this metal ion will tell us whether the copper 1 center involving other two oxidation states, that means the free metallic state and the cupric state, is present or available within the catalytic cycle. So, some physical technique, some spectroscopic technique or magnetic properties are available which can nicely detect the presence of copper in the cuprous form. So, the same thing is true, and which is sometimes very difficult, because we are handling something and we are trying to consider the corresponding molybdenum oxidation state which is within the protein envelope, because all these enzymatic systems, the molybdenum enzymes, they are coordinating to some molybdoterine part and some other donor groups within the protein pocket. So, within this protein pocket we have the corresponding center, and the sensitivity of this center towards the different spectroscopic techniques is important. So, how sensitive this particular center, it means how low a concentration we can detect if it is formed at all within the protein pocket. So, if we can go for a very low concentration, because if you have a molecular weight of several kilodalton for these enzymes and only one molybdenum center is present. So, that corresponding technique, if it is a spectroscopic technique, should be very much sensitive, such that we can detect the single metallic center which should be some kind of showing some kind of activity to the spectroscopic technique. So, one such technique is our EPR technique. So, electron paramagnetic resonance. So, electron paramagnetic resonance, if we have a unpaired electron to the system like that of our what we are going to detect for pentavalent molybdenum, which having a electronic configuration, the outermost electronic configuration it has, which is 5d1. So, this unpaired electron can be detected by the corresponding EPR technique. So, not only this particular technique, that means if we have a corresponding EPR signal, if we can detect, and if we can some idea about the environment. So, once we detect that it is EPR active if the other two forms, that means the molybdenum in the hexavalent form and the molybdenum in the tetravalent form, either they can be EPR inactive or they can show a different type of EPR signal or EPR spectrum, which is completely different from that of the molybdenum in +5 oxidation state. So, this EPR signal should be characteristic for the presence of molybdenum in +5 oxidation state, and also it can locate something the corresponding environment. So, most of the biological system, so long we do not have the exact crystal structure for the system, that means how molybdenum is bonded to the other donor groups. If this molybdenum is in 5 coordinated form, that means it has 5 donor groups and we do not know what are those, it can be A, B, C, D, E. So, initially the spectroscopic technique can detect that if you have a molybdenum in the pentavalent state, we get the corresponding characteristic EPR signal. Next, we just try to locate the corresponding environment, that means what are the donor groups A, B, C, D and E. So, if these donor groups are sometimes, if they are very specific and if they have some nuclear spin also. So, this nuclear spin can interact with the electronic spin of this molybdenum and we get some splitting. So, this particular signal what we get for this standard EPR technique is one set. So, this is the magnetic field H in Gauss and this is the corresponding absorbance. So, if we get some value, say it is 3200 or some values of Gauss. So, if this is characteristic for the center, which is the corresponding unpaired electron bearing center, and if there is some other groups, that means this particular. So, these two electronic levels, paramagnetic levels, they are getting splitted and we get the splitting around this position or in some other position if there is interaction from either A, B, C, D or E. So, one such situation is here where you have the selenium atom. So, the presence of this selenium atom is detected where we have the corresponding leveling on the selenium atom, which is 77. So, once the 77 selenium is present and which is close to the molybdenum center or attached to the molybdenum center, we can detect that this selenium ligand is present and which is showing a 77 selenium hyperfine interaction with the paramagnetic molybdenum 5 center, which is very important. So, if one of these groups, which is selenium group and is interestingly selenium 77, because in some cases also we will find that molybdenum having one unpaired electron in a pentacoordinated form. So, if you have one unpaired electron and if you have something, some species like the donor atom is nitrogen having I value something. So, that I value, with that it can go for the corresponding hyperfine splitting. So, this hyperfine splitting is originating from the donor atom, and when it is originating from the donor atom, that immediately tells us that we have molybdenum and we have nearby nitrogen also. Similarly, when selenium is bound, so if we have the corresponding characteristic hyperfine splitting due to the presence of selenium, we can immediately say that the molybdenum has some bonding interaction with the selenium atom and it is splitting the corresponding signal, which is otherwise not splitted due to the presence of other donor groups. So, this can identify that you have a molybdenum 5 center and selenium is there, and selenium, if it is not enriched with this isotope, we enriched it with 77 isotope, so that we get the corresponding hyperfine interaction. So, once we have this molybdenum in +4 oxidation state, which is oxidized to +5 oxidation state, and during that transformation we see a large structural distortion of the coordination sphere, which we are telling as the coordination environment. So, we have the coordination environment, and during the change of this oxidation state from +4 to +5, what we see that when you have the +4 oxidation state, you have one particular geometry is favored, but when it is going from +4 to +5, another coordination geometry is favored, because the bonding interactions are different and size of the central metal ion, that means the molybdenum, is also changing. Molybdenum 5 is smaller compared to molybdenum 4. So, when we have this smaller donor group, we see that this particular center, so when we have molybdenum +4 and molybdenum +6. So, in one case we have the corresponding oxidation state in +6, and in another case the corresponding oxidation state is +4. So, the corresponding demand for the electron, which is not highly oxidized, is less. So, in the lower oxidation state, we can think of the molybdenum can be stabilized with 4 donor atoms, but when it is going from +4 to +6, we expect that at least it should go for a pentacoordination or a hexacoordination, because the size is small and it will attract more and more donor atoms around it, and the bonding interaction between the center, that means the metallic center, and the donor atom is more, because this particular bond, because if we just consider when X-ray structures are known, that corresponding bond distances for molybdenum sulfur, molybdenum oxygen and other groups are different for these two different oxidation states, whether it is +4 or +6. So, always we have the preference, that means whenever we have a low oxidation state, we see that the coordination number is more or less, and when we have a higher oxidation state, your coordination number would be more. So, that immediately tells us that definitely our coordination geometry and coordination environment is changing. So, that is one source of changing the corresponding pattern of the EPR spectrum. So, if we have the central one, that means molybdenum 5 plus. So, this molybdenum in 5 plus, so if one is known as 4 coordinated and other is known as the 6 coordinated one, so it should be intermediate between these two, and that is why we get some environment which is neither the environment which is available for molybdenum in tetravalent state or molybdenum in the hexavalent state, but it should be intermediate, and while passing from this particular geometry to the other geometry, we get something. So, this is not only giving something where we have the intermediate oxidation state, but also we can have a corresponding intermediate coordination environment, and that intermediate coordination environment is surely giving us where you have a highly distorted geometry, neither the geometry what is prevalent for the molybdenum in +4 oxidation state or what we are going at the end, that means in the +6 oxidation state. So, we get something which is highly distorted. And to find this particular geometry by knowing the corresponding EPR spectrum is difficult, because we can synthesize in the laboratory large number of molybdenum compounds having some closely related coordination environment having oxygen and sulfur donor atoms can be prepared, and those compounds can be studied by EPR technique, and we get the corresponding spectrum for this particular synthesized compound. But when the environment is distorted, your EPR spectrum is also definitely going to change. So, for a distorted environment, we do not get the same type of EPR spectrum what we get for a regular geometry. So, moving from that, that means where we are moving from one particular oxidation state, that means the tetravalent state, to a pentavalent state, that means the intermediate, is accompanied by a large structural change of the coordination sphere, because we are dragging, we are attracting the 6 ligand. That is the answer to the thing what we are discussing so far. When we have the center in the low oxidation state, it can be stabilized by less number of donor atoms. When we are moving towards the high oxidation state, you are putting more and more donor atoms. So, if the molybdenum 4 is stabilized in 5 coordinated environment, definitely molybdenum 5 will prefer to have another ligand, which is our 6 ligand, and in some cases we will find that initially it is loosely bound to the water molecule, and that water molecule after deprotonation is giving us the hydroxide ion. So, when we move from this, the extra ligand is getting attracted. Since everything whatever we are talking about is in the protein environment, in aqueous environment, so water molecules are there. So, all these water molecules with their lone pair of electrons, it has 2 and 2 4 lone pair of electrons. So, initially these 2 lone pair of electrons are donated to the molybdenum center, and this molybdenum center is showing some acidic property. That means, this interaction, if this interaction is strong enough, then deprotonation can take place, and this bound water molecule can go from neutral H2O to anionic HO minus. So, we expect that this is the 6th ligand which can go and bind to the molybdenum 5 center. So, we have a dual challenge for that. Initially, the highly distorted one, and EPR should detect the corresponding distortion once we are moving from this molybdenum plus 4 to molybdenum 5. And secondly, due to stabilization for the molybdenum 5 state, we attract one extra ligand, and the very good ligand available in that environment, that means the aqueous environment, is water molecule. So, some new interaction is started playing into the operation, that the bound HO minus, that bound hydroxide anion, because this bound hydroxide anion is the main thing for all these molybdenum and tungsten bearing enzymes, because this is ultimately going for the metal oxo species, and that is the main part for the all different types of oxo transfer enzymes. So, we get from there the oxo transfer enzymes. So, these oxo transfer enzymes are definitely the core point, therefore, because once we establish this molybdenum hydroxide bond, further deprotonation, if again one H plus is moving, because here one H plus is moving, if in the second step another H plus is moving, we get a molybdenum oxo species. So, that molybdenum oxo species is the main point for the oxo transfer enzyme, and if we are lucky enough to prepare several of these oxo compounds in the laboratory as model compounds, so they basically serve as very good model species to study for the reactivity say in terms of a good example for the corresponding EPR spectra. So, we can have model complexes in our hand and not only this particular center what we are talking, because slowly we are moving from one system which has discussed elaborately is that M is our molybdenum. So, next just slowly we are moving from molybdenum to tungsten. So, tungsten will also have some affinity to give you the tungsten oxygen bond. So, other donor groups are available, and we also see how the corresponding 3D analog of this group, that means chromium, molybdenum and tungsten, and chromium also we know that the chromium is also well known for forming the corresponding chromium oxygen bond, and some of these species where we have the vanadium oxygen bond. So, all these basically, because all these metal centers, all these metal ions have a typical tendency to activate, they are basically a general thing that in all these cases we are not considering the corresponding polymerization reaction, because once we consider the polymerization reaction, what we have seen earlier that instead of this oxo we can have a dimeric species also, where you have the molybdenum oxo molybdenum thing. That means, it is the corresponding competitor for the mononuclear molybdenum oxo form. So, if one mononuclear molybdenum oxo form can react with another another non-oxo form of this molybdenum, and if this oxo can attack that molybdenum, we get the molybdenum oxo bridge dimeric unit. So, but on all these cases what we are discussing is our mononuclear system. So, in the mononuclear system you have the metal bonded to the oxygen in the double bond. So, it can be tungsten, it can be chromium and it can be vanadium. So, this particular thing, that means when we have the insertion of the 6 ligand, so it can go from water to hydroxide ion, and that hydroxide ion after deprotonation can give rise to the corresponding oxo form. So, this formate dehydrogenase is where you can have the selenium. So, the biological strain is Escherichia coli, having the molybdenum guanine dinucleotide. This is the typical cofactor. So, that is a very big cofactor what we have seen in some of our previous classes, that it has sulfur donor atoms, is the bidentate sulfur sulfur ligand. So, this cofactor is nothing but a bidentate SS donor unit, and this bidentate SS donor unit, when they are related to or closely associated with the selenocysteine residue in the polypeptide chain, our molybdenum center present in formate dehydrogenase will start interacting with the corresponding selenium atom, and if that selenium atom is labeled, we get the corresponding signature for the selenium interaction to the molybdenum site. So, if we have, because what we are looking at, we are looking for the formate oxidation. In last class what we have seen that during this oxidation, that means the formate oxidase activity, we are producing corresponding carbon dioxide. So, now if the formate is leveled with exclusively by 13C, so C13 leveling of the formate give us some idea that what is the typical source of this carbon, whether that carbon is only originating from the substrate formate, utilizing the water which is present over there. So, that is the water where this water is bound to the molybdenum center, and that is initially converted to the molybdenum hydroxide species and then molybdenum oxide group. So, this water enriched with 18 oxygen, and if it is catalyzed by this formate dehydrogenase, the gas what is produced there is only 13 CO2 without leveling of this 18 oxygen. So, no O2 O18 leveling is there. So, during that transformation, what is getting established that for all other type of reactions, what we have seen, the water is present and water is basically giving the corresponding molybdenum oxoform, and that molybdenum oxoform giving rise to the corresponding OL 18 leveling, because we are using oxygen 18 enriched water. But if the gas formed in formate dehydrogenase reaction has no oxygen leveling, we can conclude that it is not a member of that class of molybdenum enzyme for the other sort of oxotransferases like xanthine oxidase and others, where oxygen is incorporated from the water into the product. So, if any oxygen is transferred to the substrate, that oxygen is basically originating from the water molecule which is available in the reaction medium. So, this molybdenum center is therefore an unusual molybdenum center which initially coordinated to the reduced form of the molybdenum in a square pyramidal geometry. So, what we see now that we are discussing that molybdenum in the lower oxidation state can have low coordination number as well. It can have only 4 coordination number, or it can have a coordination number of 5, and the geometry is square pyramidal in nature, where we can have 4 equatorial bonds. So, 4 equatorial bonds and 1 apical bond. So, 4 equatorial or basal bonds basically. So, those 4 positions are occupied by diethylene like sulfur groups of the pterine family, and one of the other point is either attached to the oxygen or the selenium, and the diethylene sulfur atoms form a pair of pterine cofactors and selenium atom from selenocysteine 140 residue. So, if we can have this particular type of environment where we have the molybdenum and 2 of these sulfur groups. So, what we are just looking at the corresponding simple EPR spectrum, but when we have this enzyme, so directly this enzyme is available in our hand and we can detect the corresponding molybdenum center as the pentavalent molybdenum center, and it shows a typical EPR spectrum at 100 Kelvin. So, we get the spectrum when we have the thing, that means in the frozen condition, mostly the liquid nitrogen temperature which is 77 K. So, in the frozen condition, so this is the source for the formate dehydrogenase. So, this particular one is showing a signal for that EPR which is a little bit complicated in nature. So, instead of one signal, what we just have seen, which is the axial one, and this axial one can be splitted into 3. So, here if we have one G value, here we can have 3 G values, G1, G2 and G3, and that depends upon the environment. If the environment is very much symmetric and all the 3 directions, the x, y and z directions are symmetric in nature, we get the corresponding signal having 1 G value. But if all the 3 directions, that means x, y and z are different, we get 3 G values. So, we have the characteristic G1 value, characteristic G2 value and the characteristic G3 value. So, this particular signal we are discussing, only one example is taken from this paper, and this particular signal basically, so as p-paired enzyme after 30 minutes of reduction with this formate anion. So, it is basically the center where you have incubated with the formate anion, and this formate anion is giving rise to the generation of the carbon dioxide. But we see that this is one signal and this is the second one and this is the third one for a type which is G1, G2 and G3 type. this G1, G2 and G3 type basically giving rise to 3 different G values instead of 1. So, we get a molybdenum 5 species which is obtained in the presence of the inhibitors, nearly axial EPR signal showing only one detectable splitting given by the nuclear species with I is equal to half. So, when we get a corresponding axial splitting, we do not expect that there is a splitting in these 3 different values, but when there is splitting we get 3 different G values. So, this particular signal basically can support something where the EPR data is given us for not only the center, because is EPR active. So, activity for EPR spectra immediately tells us that the center is molybdenum 5, which is pentacoordinated. It can be a regular square pyramidal in geometry or it can be a little bit distorted one, and it can be formed only in the presence of inhibitors. These inhibitors, that means the azide and cyanide, so they are basically attaching to the molybdenum center, and that center, that means if we consider that N3 minus and Cn minus are inhibitors for what? Inhibitors for the corresponding reaction, the catalytic reaction what we are looking for. So, if we consider that is going from some intermediate, so intermediate is involved, which is our molybdenum 5. So, this molybdenum 5 is intermediate and which is going for completion of the catalytic reaction for the degradation of the formate anion. But if we want to stop that particular reaction, we use these as the inhibitors, and they basically go and bind, because both azide and cyanide has the tendency to bind to the molybdenum side and stabilize this +5 oxidation state. To get the corresponding EPR signal for the intermediate, we use some reagent which are the inhibitors, which can stabilize the molybdenum in +5 oxidation state, and we get the corresponding EPR signal for the molybdenum 5. So, these inhibitors are therefore important to have. So, now we just, with all these information, the spectroscopic thing for the molybdenum, we can go for the corresponding change in the metal center from molybdenum to tungsten. So, these tungsten enzymes can be very nicely compared. So, we will just compare from the molybdenum side, because for molybdenum we have studied in detail what are the environment and how they are reacting and what are the corresponding formation of the oxo, hydroxido and sulfido form. So, only thing that we can use there, that this tungsten, so we can compare this with that of your molybdenum. So, coordination chemistry of tungsten and coordination chemistry of molybdenum is very important to know. So, the coordination chemistry of these two metal ions are important to know. If some of these ligands are utilized for stabilizing the molybdenum side, whether the same group of ligands can be used for binding the tungsten and stabilizing that tungsten in their corresponding oxidation state, that means the +4, +5 and +6. So, tungsten is known for us for several years during the last decade, basically is a very recent development, and not so many enzymes whose excess structures are known has been determined so far. But interestingly, having a Z value of 74, which is quite heavy, showing some biological function, but it is not available in different eukaryotes, but it is only available in bacteria. And if we know these biological systems, then we can expect someday or other the corresponding biological activity can be available with the eukaryotes as well. The first class of enzyme for that is that corresponding oxidoreductases. They use again like that of your molybdotyrine family of ligand system. Here also we can call it as a tungstotyrine, because the same organic part is present, but which is very much available for tungsten binding. That is why they are known as tungstotyrine complex. And one such example for this corresponding catalytic activity centered on tungsten. So, the tungsten metal center in some oxidation state, they are available for reduction of carboxylic acid to aldehydes. So, if they are showing some oxidoreductase reactions, so oxidoreductases can be utilized for oxidation of aldehyde to carboxylic acid as well as reduction of carboxylic acid to aldehydes. That means, like molybdenum, tungsten center is also very much suited or well suited to interact with the carboxylic acids and aldehydes for oxygen donation or some carbon dioxide elimination from the center. So, like molybdenum center, these enzymes are also mononuclear with pterine in dithiolate coordination. So, we know that the sulfur-sulfur coordination is available and pterine backbone is there. So, this pterine backbone having in substitution. So, what we immediately write that we have this S and this S. So, these are very good SS type of ligand. So, these SS type of ligand, so once we write this in the form, so these are very good bidentate dinegative ligand and they bind to the tungsten center, and this tungsten center either in the +4 or +5 or +6 oxidation state can stabilize very well all these oxidation state, because it has electron density, more electron density. This is doubly negative in nature, is not a neutral ligand, and sulfur having this particular bigger atom compared to oxygen. So, its polarizability is also more. So, when you have a bigger polarizable donor atom compared to oxygen and nitrogen, sulfur is bigger. So, polarizable bigger donor group can stabilize the high oxidation state very much, because these sulfurs, if they are neutral in nature, they cannot donate that much electron density to the molybdenum center, but when they are in the thiol form, they donate electron density efficiently to stabilize the higher oxidation states of tungsten and also can for the molybdenum. And this particular in backbone, we are having the in backbone also. So, if this in backbone is there, so that in backbone you have giving us a very stable planar geometry. So, this in backbone is responsible for giving us a planar geometry, which is also needed. So, it basically restricts the environment, what we are just discussing, that when we have the distortion. So, if we have the tungsten environment and if these two are there and these two groups are the sulfur groups, this is one sulfur, this is one sulfur, this is from one ligand and another ligand. So, tungstotyrine unit, and we just show this double bond here, we just show this double bond here. That means, this particular basic unit. So, if we have, so if we consider the in unit, so if we have one carbon here, another carbon here, this is also one carbon here and another carbon here. So, basically this particular unit 2 plus 2 plus 1, so is a 5-membered ring. So, this 5-membered ring and this is also another 5-membered ring. This 5-membered ring supports the corresponding planar geometry. So, when two more groups, that means one group from here and another group from here or from the bottom is coming into the picture, the distortion is taking place within the entire planar geometry. If it is a reduced one, the 5-member ring can be puckered, like that of our benzene and cyclohexane comparison, because we all know that if we have a benzene ring and the cyclohexane ring, so this benzene ring, due to the presence of the double bond, is always try to retain its planar geometry when it is in the part for the protein chain or any other organic molecule, but if it is a cyclohexane one, it can have different geometries. One such is the corresponding chair form and another is its corresponding boat form. So, in 3 dimension they can go for the different type of distortion if the backbone is made up of cyclohexane instead of benzene. Similar thing happen here also, that when the backbone is made up of saturated SS unit, we get one kind of distortion, and if the backbone is made up of ene, you have one type of distortion, because the entire planar group, because the planarity should be retained, this planar part, this planarity should be retained. So, the entire planar part would move around this tungsten environment such that you can accommodate the new donor group. If it is initially pentacoordinated, we have to put the 6th one. So, this particular group is moving to accommodate the extra coordination, and the entire planar unit is moving simultaneously to accommodate this extra group. So, this terene in dithiolate coordination is therefore important. So, in backbone is giving us the planar geometry, and the protein crystallography for these, so X-ray crystallographic results of these several enzymes, that we can add the corresponding oxo groups to it, to the tungsten center. So, it can be one oxo center or it can be 2 center, because already we have seen the tungsten environment is covered by 2 of these terene units. So, the remaining 2 positions, the coordination number 5 and coordination number 6, can be occupied by one oxo or both the oxo groups, can be occupied by sulfido function, cysteine function, selenocysteine function. Serine is giving the oxygen from the amino acid residue, or ACO group, when nothing is available, the water molecule is bound to it, or followed by a deprotonation of the water molecule giving rise to hydroxide coordination, and or a second dithionyl in various metal coordination spheres. Or we can have, we do not have the corresponding option for binding these oxo and hydroxide groups, but we can bring the 3rd dithiolane unit also, because we can accommodate a hexa-coordinated environment around the tungsten site. So, due to that, and we are just looking for electron transfer reaction, because all these reactions based on tungsten center, we must have electron transfer property. So, the redox flexibility, which is a very important terminology for that, that if we have the corresponding oxidation states like molybdenum, that 5 plus, then molybdenum 6 plus and 4 plus. So, how quickly we can convert without much distortion, without much reorganization, how quickly we can interconvert between these 2 coordination geometries, because if there is a tremendous barrier for the change in coordination environment, we do not get facile or reversible electron transfer. So, always we get, or always we expect a very good redox flexibility in this regard, because as we go for the corresponding oxidation from this to this, immediately the coordination environment should be adjusted, or as we move from +5 to +6, the coordination environment should be adjusted. So, when we go for something, that means we go for a corresponding cyclic voltammetric measurement. So, the CV, the cyclic voltammogram, what we basically get. So, if there is no such reorganization, so this is for the oxidation. So, we scan it from left to right, and if this is for oxidation, we are producing from something where we have tungsten 4 plus is under there, and we are producing tungsten 5 plus. So, there would be some interconversion between these two oxidation states, and this particular technique is very useful to know that how much this particular oxidized form, that means the tungsten 5 is stable. So, we switch back, the scanning is reversed such that we get a corresponding signal for the oxidized form. So, this is basically tungsten 5 plus is reduced back to tungsten 4 plus. So, if reduction is facile, redox flexibility is available, we get there that this particular peak is there. Otherwise, what we get, that means that 5 is there and 5 is not so stable and it is undergoing some decomposition. So, the trace would be like this. So, this trace will definitely go for in different direction, and we are not expecting that tungsten in plus 5 oxidation state is stable there. So, this particular flexibility of this metal ion can be coupled to proton as well as electron transfer. So, in one direction, if we have the electron transfer, so electron is taken out from the system, and if we are doing this, that means the electron transfer is taking place in corresponding aqueous medium, in water medium. So, proton transfer will take place in the reverse direction. So, if the electron transfer is taking place, that means we are taking out the electron. So, if we have one electron loss from one direction, we can have the corresponding proton transfer in the opposite direction, and we call the both of these two are engaged in some equilibrium, which we call as electroprotic equilibrium, which is known as electroprotic equilibrium. And this particular thing is also very much important, not only for the molybdenum center or the tungsten center, because we are basically looking for the corresponding coordination of this molybdenum or tungsten with that of our water molecule. So, it immediately tells us that how good these things are for these informations, that this particular one, when we go for this oxidation, that means plus 4 oxidation state is moved from plus 4 to plus 5, and center is becoming much more electron greedy, because we are taking out electron from the center. So, during that electron transfer, that means when we are doing for cyclic voltammetry, the electrode is taking the electron, or if some other oxidizing agent is present. So, that oxidant is taking that electron from the center. So, at the same time, this particular water molecule bound to the center will have the tendency for deprotonation. So, this particular one is removed from there as H plus. So, this is our source of proton. If it is not coming from the other side chain, from the enzymatic side chain or the polypeptide side chain or the amino acid side chain, it can directly come from the water bound to the center, and which makes it this particular center as the hydroxide of function with a corresponding removal of the proton from the system. So, if the same center is moving from 5 plus to 6 plus, and we have a bound water molecule. So, again our electron transfer can take place, So, this particular one is going. So, if this goes as H plus, like another step of proton transfer, and this one is basically this electron density is donated to this, and we get a tungsten oxo form. So, due to those electron transfers from the systems involved, and particularly this particular the removal of this proton from the water molecule, we get the corresponding tungsten oxo form, and this oxo is utilized for a very good substrate or very good reagent for all these enzymatic reactions where oxo transfer is taking place. So, during this redox transfer or redox transformation, we have the coupled, that means these two are indistinguishable. Whenever you have electron transfer in the system, you should always think of that there is a proton transfer, and this oxo transfer mechanism what we know for the corresponding oxidation related to xanthine and other. So, for the tungsten enzymes also, once you make the oxo form there, that oxo group can be donated to the other substrate molecules involved for the oxo transfer reaction. So, slowly, like molybdenum enzymes, so far not so many tungsten enzymes have been characterized in detail, and during the last say decade or 15 years or so, some number of tungsten containing enzymes, we call it as W enzymes, the tungsten bearing enzymes, are hyperthermophilic from archeria, because they are stable in high temperature region also. So, at high temperature they are very stable, and our most of the cases our notion is that all these biological system, all these enzymes are not so stable at high temperature. So, this thermophilic bacteria, so they can stabilize at higher temperature also. So, these forms, that means the oxo form of these tungsten bearing enzymes are getting stabilized, though they are also bound to sulfur groups, because sulfur groups are not so well known for its stability at higher temperature. So, largely speaking, we can have three classes of these tungsten enzymes. So, these three classes of tungsten enzymes, one is the Formate dehydrogenase, what we have seen in case of molybdenum also, that this Formate dehydrogenase is also there. Then aldehyde oxidoreductase. So, aldehyde should be oxidized to corresponding carboxylic acid, and that particular electron transfer is all the time coupled with iron sulfur proteins. So, we have these iron sulfur proteins, that means the ferredoxin molecules, and these ferredoxin molecules are responsible for electron sink, that means they can take off electron from the system and they give up the electron to the system. In such a way, the substrate aldehyde is getting oxidized. And another form, so this is aldehyde ferredoxin, and which is particularly well known for formaldehyde, because formaldehyde, though it is a aldehyde, but it has some different property, because you have the corresponding carbon hydrogen bond, and that carbon hydrogen bond has to be broken for the corresponding reactivity of the formaldehyde to formic acid. And there also the ferredoxin iron sulfur proteins are involved, and these ferredoxin iron sulfur proteins are responsible for electron acceptance. So, next day we will just continue from here. Thank you very much.